All right, hello everybody, and welcome to the June NITheCS Mini-school. Today we have lecture 1. We will have 3 other lectures on the 11th, 18th, and 25th of June. Today I am standing in for Prof. Francesco. I am Donovan Slubbert, one of his PhD students. And June's Mini-school is about monitoring biodiversity using data cubes, techniques and applications for open science. And today's lecture is titled Introduction to Data Cubes, Fundamentals and Applications. Gain a foundational understanding of data cubes, their structure, and their applications in biodiversity monitoring. This lecture will provide essential skills and knowledge for sustainable development. Presented by both Sandra, MacFadyen, and Maarten Trekels. I'm not sure how you guys are going to handle switching over. I'm assuming, Sandra, you'll be first, or you're muted at the moment, Sandra?
Yes, I'll start off, and then Maarten will take over midway.
Okay, perfect. All right. Then, Sandra, please feel free to start, and then we'll be here listening. Thank you so much.
Great, thank you. Can you all see my screen? I should have shared it by now. Okay, perfect. Thank you. Thanks so much, Donovan. As Donovan's already said, welcome today to the first session of this Monitoring Biodiversity with Data Cubes. I'm Sandra MacFadyen, and together with Maarten Trekels, we will explore how data cubes can transform the way we monitor biodiversity and biodiversity change. Just a bit of background about ourselves. I'm a research fellow with the Mathematical Biosciences Lab, Biomath, we call ourselves, at Stellenbosch University. I'm also a NITheCS work package leader for research focused on ecological complexity and biodiversity, and a member of the EU-funded Biodiversity Building Blocks for Policy project. And that's where the data cube component comes in quite vividly. I trained as an ecologist, and I've worked for many years with South African national parks before pursuing my PhD, so it was a bit of a late start to my PhD studies. But I continue to live and work in the Kruger National Park, and as such, my interests lie a lot in macroecology, ecosystem dynamics, and applied spatial temporal statistics for biodiversity conservation. Maarten is a PhD student in our group, supervised by myself and Prof. Kang Hui. But I'll hand over to him to give you a little bit more info about himself. Maarten? I can't hear you, Maarten. I'm not sure if the others can. Still not.
Can you
hear
me now?
We can hear you now.
Yes. Okay. Sorry. So as Sandra said, I'm a PhD student in the Biomath group at Stellenbosch University. But actually, my background is in physics, and after a while being in industry, I moved about 6 years ago to the biodiversity informatics sector at Meissen Botanic Garden in Belgium, where I first worked at projects related to the DISCO research infrastructure. DISCO is the Distributed System of Scientific Collections, where I was mainly looking at data standards and how to develop these data standards. And then moved further on to more the data cube related projects like biodiversity building blocks for policy. In the meantime, I'm also a member of the Research Data Alliance, where I look at fair data and interdomain interoperability. So back to you, Sandra.
Thanks, Maarten. All right, so just to give you a quick overview of what we plan to do in the next few weeks, we'll introduce you to the idea of data cubes as a scalable and open source solution to address global biodiversity challenges. And each Wednesday we'll take you through different sessions. Today's session is, as we've said, the introduction to the concept of biodiversity, as well as the idea of data cubes and how we can use those to monitor biodiversity. We'll touch on different sources of biodiversity data and talk about the data standards that need to be applied to use those data. And then we'll discuss briefly data publication and fair data publishing principles. We'll end off today with a little bit of homework for you. You'll need to set up 3 different free accounts for you to continue with the different sessions. Today is more theoretical, but then the next 2 sessions will be very hands-on. So we'll be doing some coding in Colab using Jupyter Notebook, either Python or R. We'll be switching between them so that you can get a hands-on feel for how to work with these data cubes. If you have your own data that you would actually like to test these different principles, even better. So if you've got a tabular data set with latitude and longitude coordinates and maybe a temporal domain, please bring that along, and you can start working on these things using your own data. Then the next session on the 11th, session 2, will be the hands-on session by Maarten. You'll be looking at building data cubes with GBIF data. He'll give you a brief introduction about GBIF itself, and then go on to a hands-on guide for getting started with biodiversity data cubes. I'll then lead session 3 on the 18th, where we'll dive into some ecological modeling with these data cubes. By the end of the session, you should be able to create your own data cube using GBIF data, or as I said, if you've got your own data, please bring that along. And we'll be using Google Colab and Google Earth Engine for that. At the end, you'll also be able to visualize your results on an interactive map through these interfaces, and you'll be able to analyze some basic patterns of species diversity related to environmental variables like climate, for example. Our last session on the 25th will look more at the concept of open science and how you can use different methodologies to share and disseminate your results. We'll have a bit of a hands-on session for that using GitHub and Zenodo. So if any of these phrases are unfamiliar to you, we will be going through them throughout this course. And then we'll wrap up after session 4 with some inputs from you. We always like to hear if you have other ideas for different mini schools later on. This is the second time we've presented this one, but we're open to presenting different ones. So we'll give you a questionnaire to help us fill that out. So if we dive straight in, today, as I said, is lecture 1, it's just an introduction to biodiversity and also biodiversity data cubes specifically. Maarten and I will co-present this session. And what normally happens is while I'm presenting, Maarten's manning the chat. And when we switch over to Maarten, I'm manning the chat. So if you have any questions throughout the presentation, please feel free to pop it in the chat. One of us will answer. Otherwise, at the end, we'll take questions as well. So if we go straight into what is biodiversity, it's essentially the variety of all life on Earth. But what we often forget is it includes the ways these different organisms interact with each other as well as their environment. So to put this into context, all species, plants, animals, and humans, depend on each other to survive. So that includes us. So the loss of biodiversity leads to a loss of services that nature provides that are essential to the functioning of our society and our economy. Services like natural processes, nutrient cycling, for example, resource provision, food and water for us and other animals, regulatory systems, and of course, cultural benefits. Being in South Africa, everybody loves the Kruger Park. Everyone enjoys protected areas. And it definitely has an intrinsic value to us without having to put a monetary value to it. So in other words, biodiversity loss directly threatens ecosystem health, the services they provide, and then ultimately human well-being. And recognizing the importance of this, the Sustainable Development Goals were developed in 2015 by the United Nations as part of the 2030 Agenda for Sustainable Development. These goals aim to protect biodiversity by promoting sustainable use of ecosystems, conserving species, and ensuring that human activities do not harm natural habitats. And in response to this, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystems, commonly known as IPBES, this was formed to assess the status of our biodiversity globally, its conservation, and develop sustainable use goals for their use. Many of the IPBES assessment reports that came out of this initiative make it very clear that more efforts and comprehensive actions are needed to halt biodiversity declines. It's happening globally, and we need a way to manage it. So to recap, biodiversity is the foundation of healthy ecosystems. And if we're in the midst of global biodiversity decline, it's because of things like habitat loss, climate change, and other factors that we will touch on throughout this course. But how do we respond to such a global crisis? The simple answer is we need global standardized solutions. In other words, if biodiversity decline is our major concern, we need a scalable approach to measure biodiversity change across different spatial and temporal scales. But in order to do this, we first need to understand the concept of biodiversity itself, and that's what we'll dive into today. So we've already established that biodiversity is the variety of life on Earth, how that life interacts within and with their environment. But it also has different scales to biodiversity. I think most of you, when you think of biodiversity, you think of species diversity, and that's simply the variety of species within an ecosystem or across the planet. But before that, we have something like genetic diversity, which is the variation of genes within species, allowing for adaptability and resilience within that. And then we have something like ecosystem diversity, which is the variety of ecosystems that provide different habitats and different services to different species. And as I think you can see, it's already very interconnected, this notion of biodiversity. But it doesn't stop there. To add complexity to the system, there are also different components to biodiversity, each with their own scales. And in most cases, when we think of biodiversity, I think most of us think of compositional diversity, things like species diversity, the types of species. And those scales are represented by the different scales, as I said, genes, populations of species, ecosystems and communities, and different types of landscapes. But then we also have something called structural diversity, which has a similar set of scales that we walk through in terms of gene structure, population structure, ecosystem or habitat structure, and landscape patterns. And the third tier is what we call functional diversity, which is essentially ecological processes that boil down to genetic processes, population dynamics, intraspecific interactions, and landscape processes. So you can see it's a very nested concept with different scales, that I think you can already start to believe that it's a complex entity on its own that we need to try and unpack. So if we think of a landscape, for example, a protected area landscape like the picture I've got here on the right of Kruger, one of the rivers in the south here, we first need to decide on the domain or the template across which we plan to actually quantify biodiversity. So it's the variability of different elements that we've spoken about and their interactions. But that variability is happening over space and time. So we could put in space as an x-axis and time as a y-axis, and then our different variables would be the third dimension of that. But we need to get to that pattern that we see on the top right there. And as we start to add the different elements that make up our landscape, we could begin with something as simple as the underlying landscape elements that form a more stable template, something that changes only happen over long periods of time. Things like topography, geology, soils, climate, all of those factors or elements that create the different patterns that we see and drive the processes. On top of this landscape template, vegetation can begin to establish and interact. So this leads to things like vegetation dynamics in terms of grass, shrubs, trees, but not just the composition, also the structure and the function, as I mentioned before. We have to keep that in mind throughout each of the elements that we work with. Animals are likely to come next, now that they have the basic requirements to survive, things like food, water, and shelter. And that brings in a whole other dimension of dynamics. Animal dynamics, like species composition, trophic interactions, etc. Sorry about that. All right, so at the same time, we mustn't forget about disturbance events that may alter stable system states. So all of these elements that we've added together. Those are things like fire, floods, droughts, human activities, things that are natural and unnatural in the system. But there are shocks to the system that might change the patterns that we would expect to see under normal circumstances or conditions. And then we've got the more complicated pattern and process feedback loop that we're seeing. Once we have these individual elements added in at a specific spatial and temporal scale, they start to interact. And a simple example of such a landscape pattern and process feedback loop could be something like elephants in Kruger. Elephants would typically knock down large trees. They would open up the canopy. This would allow more grass to grow. And in turn, because of higher grass biomass, the fuel load is higher and fires will become hotter and thus burn smaller shrubs, so that trees will struggle to recover from that environment, essentially keeping it in a grassland state. And there are a number of different examples of this. Birds eating fruit, dispersing the seeds in their droppings. New shrub patches then form, which then create more fruiting plants, which create more habitat for those specific birds, and so on and so on. I can go on forever, but it's important to know that these interactions are happening. So how do we measure this? And data cubes offer a scalable open source solution that allows us to capture the complexity of these different components of biodiversity across different scales. So we'll unpack this a bit further and look at data cubes specifically in the context of biodiversity. And we need to first understand, how can data cubes represent biodiversity? So they are essentially multi-dimensional arrays of data that are used often for spatial temporal analysis because of the dimensionality. And those dimensions and structures can be broken down into at least three components. The first being time, as I've mentioned, space, and then a variable or variables of interest that you're going to be adding to that. So in its simplest form, a data cube can comprise the value of a variable of interest, for example, rainfall, over time, could be over years, months, days, and across space. So you can have a latitude and longitude representing the extent of Kruger, for example. So we can easily visualize this, or the temporal trends of this at least, with a simple graph that shows us how rainfall might change over time. We could take that and display it spatially by producing a map of long-term mean rainfall that shows us the pattern, the spatial arrangement of rainfall over the long term. But there's a spatial temporal link that is often ignored, where rainfall in this case is variable over time and space. And when we're considering biodiversity, and as I said to you earlier, the interactions that are happening all the time, we need to also consider how different variables will respond to rainfall and how they will interact with each other as a result. For example, the interplay between grazers and browsers as rainfall influences that habitat. And when quantifying these patterns, we must not forget that our interpretation will be scale dependent from a space and a time perspective. So for example, if we're interested in the spatial variability, is it at a global scale, a regional scale, a local scale? What is the finest resolution that we're trying to investigate this variable at? And then time. Are we interested in changes that are happening every hour, every day, every month, every year, so on? It's important to choose the right scale for the ecological question of interest, and that's something that really needs to be considered every step in the analysis process. And identifying the correct variables as well as their interactions is another important factor to consider. And by adding more dimensions to our data cube, we can account for interactions between things like genes, species, communities, and their environment, but this adds complexity. So data cubes can represent this variable over time and across space. And by combining variables into multi-dimensional matrices, this allows us to also track these interactions, which leads to the dynamics of environmental change that you can visually see in many cases. So if we think of our initial landscape that we had previously and the patterns that we see there, we need to remember that these patterns are formed or maintained in response to the composition, structure, and function, including interactions of and between a number of different variables like landforms, rainfall, temperature, plants, animals, all the different things we've already discussed. But how do we quantify this? How do we bring this all into a data cube setting? So we can begin by unpacking which are the elements we suspect will cause and or affect the patterns that we see, and then quantify those over space and time. For example, we can look at variations in rainfall, temperature, plants, animals, and this would easily be captured in a simple matrix or a 3D structure, the one that I showed you earlier with your X and your Y and your Z axis. But to visualize this in the form of a data cube, we could take just one variable, for example, rainfall, and numerically represent one time point, for example, year 1, as the data frame or the matrix, with a latitude and a longitude coordinate and the value of that variable. And by adding multiple time points with the same latitude and longitude, we can essentially create a data cube. In other words, each dimension represents a new time step, and the cells the measure of interest at a specific location of a specific scale. And we can do the same for multiple variables, adding these different variables to our biodiversity framework. We can add multiple dimensions to our data, creating what is a stacked matrix or a data cube. However, we can't just add these all up. So once we've got all of these spatial temporal slices of different variables, we can't just add them up to get back to our initial landscape or the biodiversity pattern that we're investigating. So biodiversity is greater than the sum of its parts. And we need to always keep in mind that ecological systems are dynamically complex and adaptive. So for example, there's also variability in the ways how different variables interact over space and time, making it extremely complicated to understand different outcomes. Yet this understanding is essential for effective biodiversity conservation. So data cubes are our best bet for tracking biodiversity and environmental change across scales. But we need to have reliable, accurate, and repeatable data and methods to create these data. So we need good data to develop sound understanding, the whole concept of rubbish in, rubbish out. I'm sure you've heard that already at primary school level. And that is that the quality and the utility of analysis and results rely on the well-structured, standardized biodiversity data. And data cubes offer a scalable approach to track this biodiversity and environmental change across scales. But to effectively use it, the data needs to comply with the FAIR principles, being that it should be findable, accessible, interoperable, and reusable. And I'm going to hand over to Maarten now, who's going to introduce you to these key concepts around the FAIR data principles, definitions and standards, and the importance of making biodiversity data specifically fair.
Thank you, Sandra. Can everyone still hear me? Okay, good. I will start sharing my screen. Maybe it's easier. So everyone should be able to see my screen now. So I will dive a little bit more into the details of biodiversity data on some definition standards and what is FAIR data. And especially because biodiversity data, and this will maybe respond already a little bit to one of the questions in the chat, has some particular properties that we have to take into account when looking at biodiversity data. So what will I cover in the next coming slides? What are these sources of biodiversity data? How do they get published? How can we find this data? Where can we find this data? How do we standardize this data? And in the end, something about FAIR data publishing. And I think it's quite important to understand that biodiversity data is really very diverse. You cannot talk about biodiversity data just like temperature data, which is already you have two formats of in degrees Celsius and degrees Fahrenheit, but at least there is a conversion in between. Biodiversity data comes from a lot of different sources. You have national checklists, which are basically lists of species that are present in an area or a country. You have citizen science, and I think the most well-known citizen science project is iNaturalist, where just anyone can go into the field with a cell phone, take images, and use the algorithms of iNaturalist to check what species it is. And there is a system of peer review of these observations that make sure that the identification is right. You have natural history specimens like the one here shown in the middle, which are historical samples of biodiversity that were collected over time all over the globe and stored in collections, but that contain a rich amount of information on the labels, or there is some genetic information still present in specimens, especially those preserved in alcohol. And you have ecological surveys. These can be done by camera trap images. You can have people monitoring plots, but you can also think of a vessel going out on the sea and trolling a net behind it and see what can be captured. So there are lots of sources of data, and they all have their own specific properties. And you can already see that biodiversity data is not something which is very homogeneously distributed over the globe. You have some hotspots that are better surveyed than others. There are biases. People tend to collect, if you look at citizen scientists, for example, collect near roadsides or near houses what the biodiversity is, but in the very remote places, data is lacking. But there are also a couple of less known sources of biodiversity, and one of them is biodiversity literature. And I'm here taking an example of Plati, which is a Swiss-based NGO that tries to extract treatments of species out of literature. And I have here an example of a species that were described at some point based on specimens that are in collections. But at least you can see that one of these literature overviews contains a lot of information on this species, where it was found, when it was found, by whom it was found, and it often also contains a description of the habitat where this species were living. And biodiversity data is not something which is on its own. So far, we were talking about more or less point data or point-like data. More and more also biodiversity is derived from remote sensing. And so you can derive coverage, but even in some cases look at species composition from outer space. So this is an additional component, and one of the reasons to ensure that our data is fair and especially interoperable with these other types of measurements that are going on. Because it's only by integrating all these sources together that we can make sure that we make the most of our biodiversity data. How is this data now published? So people are collecting this data and storing this data in databases, but actually we want to liberate this data and make them available for everyone. And there are a couple of steps that you can take to make this data available or to find this data, and that is in specific platforms. For example, on the top right here, you see the portal of our own institute or my own institution, where we have an herbarium with plant specimens. We have a portal where you can see the specimens that were collected and the data that were derived of these specimens. And there is even the possibility to download this data in CSV format, for example. Citizen science projects do the same. I talked about iNaturalist. Now, the example you see on the left is a local initiative here in Belgium, which is also a citizen science platform, where you can see that people that you can find online the images that people were taking, when they were taking those images, and what is the species that was observed. This is single species. If you look at ecological data or more survey data, you can see that there are also initiatives like the Environmental Data Initiative, where you can package up your data and make it available to the public, and then people can start downloading this data. Of course, all of these initiatives are very valuable and very useful if you need a specific data set that you have in mind to analyze. But the big power to biodiversity data is when you start combining all these different sources into one single source. And one global initiative to combine all biodiversity data is the Global Biodiversity Information Facility. This organization tries to collect all kinds of biodiversity data sets together in one single platform, and then make them available already in a sort of fair way. So in this, in the Global Biodiversity Information Facility, you can find all kinds of different data sets from all sources. So in this case, not everything is like in the previous slide, where a platform is really developed for a specific use case. This one is gathering all of them. So it's not only ecological surveys, but it's also the collections and so on and so on. So here you really get a more total view on what's available on biodiversity data. And I made this figure here a while ago, where you can already see some of these biases that I've talked about. And one of these biases is where the data sets are made available. So some countries are really actively making all these data sets available, while other countries are not part of GBIF and are not making them more accessible. Another bias that you can already see is that highly densely populated areas like Europe, you have a lot of observations on biodiversity, while you almost have none in the Sahara Desert, for example. So these are biases that you really have to take into account when you look at the data and when you start try to make sense out of these. We will get a closer look to GBIF in the next lecture, because when we will start building the data cubes, we will start off with the GBIF platform, and in that sense, I will dive a little bit deeper into how GBIF is structured in the next lecture. But one of the good things of publishing your data into GBIF is that GBIF has a mechanism in place to track how your data is being used in all kinds of different publications. And it also makes your data more valuable. So, for example, here there was a publication in 2021 that was looking at how GBIF data was used. And you see that about 2 papers a day are published using this GBIF mobilized data, and not in the fields that you initially would expect. So you see here is a little graph of where all this data is used and in what kind of keywords were used when looking at the data. And you see that species distribution models are very prominently present, but also functional ecology, some people and nature, even niche dynamics and so on. So, or diseases, for example. So actually, by opening up your data and making it interoperable with other sources of data, you open up a whole different kind of world and make it valuable for other fields as well. We talked a lot about fair data principles, but I think it's now time to just dive a little bit deeper into what are the fair principles. And as Sandra mentioned before, FAIR stands for findable, accessible, interoperable, and reusable. So I will mainly focus today on the findable and the interoperable section, but we will try to cover them all a little bit. Let's look at findable. And I think to have something findable, you need to publish it in such a way that it's easily retrievable according to a certain protocol. And I think the most well-known way to find, for example, a publication is the persistent identifier that is attached to a publication. And the most well-known persistent identifier is a DOI. This is a persistent link that always works and always sends you to a landing pages of, for example, a publication, but also data which is derived from GBIF will be assigned a DOI so that it stays findable in the future. DISCO, the project I'm working on here in Belgium, also tends to assign these DOIs to individual specimens to make sure that these specimens are easily findable. If we look at the interoperability, interoperability means that if you get the data or if you see the data, you immediately have to know what's in that data so that you don't have to think about, oh, what does this mean? What is this particular field in my data? How should I interpret this? Interoperable means that you use data standards and vocabularies that are structured and curated by a community, such that actually whenever someone uses these fields, it's immediately clear what it means. And I started my experiences in the biodiversity informatics by developing standards within the Biodiversity Information Standards Organization. So in biodiversity, it's like you have the ISO standards in some technical fields, or there are lots of other types of standards around. Think about a very simple one, which is PDF. PDF is a standard that if a machine gets that PDF, it knows what to do with it and how to display the data which is in such a PDF. Biodiversity Information Standards tries to develop these standards specifically for biodiversity data, and it's a community effort. So basically, anyone from the biodiversity information community can engage with this organization and start developing standards. And one of the most known standards is the Darwin Core standard. The Darwin Core standard is the standard that describes an occurrence, or at least one single occurrence of a species. And it has a few terms that are defined. And by reading those terms, and I don't know, but I have an example here. It says event date, for example. Event date is one of these Darwin Core terms. It has a definition to make sure that that you know what is exactly meant by event date. There are some comments, and for example, the comments can be that it's actually expected that this field conforms to another standard, and this is an ISO standard. And some example use of this field in different types of situations. So that in any case, if everyone uses in his data set the term event date, it's exactly known what is meant with event date. Of course, every database or every researcher, every research question will have different databases with all their data, sometimes covered in CSV files or in other types of files. So there is always a mapping effort that needs to be done. You cannot do a one-to-one relationship to your to the data you have lying around to this new to the Darwin Core standard, for example. But the Global Biodiversity Information Facility has a toolkit that helps you mapping your columns to specific fields. And this is the IPT. And if you ever get in contact with this, there are ways that help people map their data to this same terminology. And there are also existing vocabularies, for example, vocabularies on pollination, different terms, that standardized terms that can be used to describe a pollination event. And then the last one is that I want to highlight here is the reusability. So it's very important and very, you need to take this into account when you publish your data, is that your data only gets more value when you publish it with a clear statement on how people can reuse your data. If you don't put that statement, it kind of assumes that your data is not, people are not allowed to reuse your data. So if you publish it, you really have to put a license on it. And this license just describes what other people can do with the data that you published. And one of the most well-known licenses around are the Creative Commons licenses. You might have heard of them, and you often see these kind of symbols appearing on the publication, for example, but also on datasets. And most of these times, these licenses are well described of what you can do with the data or what you cannot do with the data. A CC0 license is actually not a real license, it's a waiver, so that means that the data can be reused without any restrictions. But one of the most used licenses is CC BY. The CC BY license really just say that you can use the data as long as you give the proper attribution, as long as you cite properly where this data is coming from. Another one that you can often see popping up is the non-commercial license. So if you ever see CC BY and C, it means that this data, you need the attribution. So the BY stands for attribution. The NC is non-commercial, so you cannot use it for commercial purposes. And sometimes this might have big implications. And you have to be very aware if you publish anything under a non-commercial license, it's often also not possible to use this data on big platforms such as Google or Microsoft Azure and so on. So if data sets are derived and put on a specific cloud provider, very often everything which is under a non-commercial license has to be filtered out. It's very restrictive, and also, for example, if you put images online of your biodiversity, whenever there is a non-commercial license on it, it can also, for example, not be reused on platforms like Wikipedia. And this is an example on how this works on GBIF. So you have here the herbarium publication of my institution. It's an old one because I see that there are only 1.8 million occurrences online. Currently, it's a bit more already, where all these things are shown, and especially here under the license, you see that all this data is published under an attribution license. So all of the data which is coming from our herbarium are free to use as long as you properly attribute the data source. And GBIF is clearly also putting that metadata visible on their home pages. So whenever you use this data, it should be clear which license is on there. Of course, we are in a very moving field, and lots of things are changing very rapidly. And so what we have to do now, and what people are doing at the moment, is to make sure that the publication of this data is becoming more and more easy. And so we do need to encourage the collection and publication of data, especially the open publication of data, because a lot of publications, people are collecting data sources, but they keep them for themselves. Of course, this data can get lost. That's why it's very, very important to publish your data into open repositories such as GBIF. We also need to develop better monitoring methods that can be cheaper and more reliable, and also can go into remote areas. Think of monitoring by drones, for example. But all these data come with their specific properties. So also, although it's something which is easily forgotten, a lot of effort needs to be done in how to standardize this data and make sure that they become available in GBIF. At the moment, GBIF is also developing a new data model that would allow us to publish more rich information. One of the nice challenges, and if anyone of the students here still wants to participate, it's still possible. Every year, GBIF is organizing a challenge, which is called the Ebbe-Nielsen Challenge, where you can develop innovative biodiversity informatics tools that help people to publish their data, or that can help analyzing GBIF data and so on. Last year, the winner was using actually ChatGPT as a way to help people get their data published in a standardized format. But this challenge is still open. I think the 2025 Ebbe Nielsen Challenge has a deadline somewhere beginning of September. So if you have a great idea to do with GBIF data, don't hesitate to try to win the Ebbe Nielsen Challenge. Another component, and one we will touch upon in next lectures, is cloud computing. So currently, lots of researchers are still downloading the data from a source or from GBIF to their local machines, do their analysis. But if you want to make interoperable analysis of this data with other data sources, and since the biodiversity data is really booming a lot with the onset of citizen science and all this kind of automatic detection of biodiversity, it becomes more and more difficult to run these analyses on a local machine. So one of the things that we will focus on in the next lecture is how can we optimize this by using cloud computing as for analyzing biodiversity data, or to make sense out of biodiversity data. And there are already some initiatives going on, and so you can find the GBIF datasets already on the Microsoft Planetary Computer, but also on Amazon storage. There is a GBIF dataset, and even in Google you have BigQuery, where also part of the GBIF dataset is stored. And in the next lecture we will use Google Earth Engine as one of the cloud platforms to do nice things with biodiversity data. Okay, so then I think we're done with the theoretical overview of this first lecture. Are there any questions, remarks, things we should explain better, or which need some attention? Please let us know. I don't know, I can't see the chat at the moment.
Yeah, I'm looking at the chat, I can see it. Nothing in there.
Okay. I think Sandra mentioned already that if you have own data sets and so on, or problems that you're struggling with specifically to biodiversity data, also in the coming week, don't hesitate to contact us. We could try to have a look at it before the lecture. I cannot promise that that's gonna work out, but at least if you have some specific problem that you're dealing with and you want an answer to, just you can post it as a question and we can see whether we can do something with this.
Yes, yeah, I'd like to reiterate what Maarten said there. It really works wonderfully if you bring your own data. If you've already got a question that you need answering in your field, bring your data with even just a CSV file with the lat and long, a time, and a variable, and we're going to show you how to bring it in. Before anyone runs off, I did put in the chat earlier, but please remember to yes, there we go. Please remember to set up these different accounts. They're all free. So to continue with the next sessions, you'll need to have a free GBIF account, a Google Earth Engine account, and a Google Colab account. So we'll use GBIF to access data in a CSV type format of species occurrences. We'll use Google Colab to start coding in Python and R, and we'll retrieve background information from Google Earth Engine, things like temperature and rainfall. So please set those up before the session. The next two will be very hands-on. And as Maarten said, if you have any problems, you can email us and we'll respond before the sessions.
Yeah. I see a question popping up in the chat. Are there any international standards for sampling biodiversity data? Yes, there are. There are well-defined protocols to do surveys. And actually there is, if you look at the Darwin Core Standard, or at least the extensions to the Darwin Core Standards, there are ways to indicate what sampling protocol you have used. Basically, there are ways to indicate I recorded according to this standard on this location and so on. But there are many standards on sampling, so I failed to get one specific example, but depending on, for example, some reporting strategies in your country, there might be a specific standard needed how to sample certain data. One thing I maybe forgot to mention on GBIF data, and it's something to also keep in mind when we are going to the next lectures, is that GBIF data, the way it's now defined, is presence-only data. So it's not covering absences. So if anyone surveyed a specific area and said, I've been here on this location for days and I didn't see it, so it's not there, that's something you cannot represent in GBIF at the moment, but they are working on this.
Okay, thank you, Maarten and Sandra, for a very comprehensive lecture. If there are no more questions, then I think we can end the session here. Please remember that next week, Wednesday at 2 p.m. again, we have lecture 2, which is titled Building Data Cubes with GBIF, a hands-on guide for biodiversity monitoring. So if there are no more questions, we can end the session here, and we will see you all next week, Wednesday.
Thank you, everyone, for coming. It was a great turnout, and hope to see you all next week where we can do some hands-on stuff. And thanks, Donovan, for setting this up for us.
Yeah,
of course. All right. Thank you, everybody. Goodbye.
Bye.
Bye.